Welcome, everyone. On behalf of the United States Aquaculture Society, the National Aquaculture Association, and the Alabama Cooperative Extension System, we'd like to thank you for taking time to join us today. As the aquaculture industry expands throughout the world, genetic considerations have become increasingly important in terms of production, sustainability, and conservation. Unfortunately, producers and policymakers often perceive genetics as a complicated and intimidating topic. As a result, they may be reluctant to delve into the subject despite its increasing prevalence. We're very fortunate today to have with us Dr. Greg Lutz to help demystify genetic improvement in aquaculture. Dr. Lutz is a professor and extension specialist with the LSU Ag Center's Aquaculture Research Station and School of Renewable Resources. He's the author of the book Practical Genetics for Aquaculture, and his career has included experience in research, teaching, and extension outreach, as well as private industry, with a focus on hatchery and production practices. And although he's worked with numerous species in several countries, his favorite topic is still genetic improvement. And with that, I'd like to turn it over to you, Greg, and take it away.
Well, I also want to thank everybody for being here today. The idea of this webinar, this presentation is pretty much what it says right there in the title. It's the basics. You can make genetics as complicated as you want to, and there are a lot of people that write books about it. We're going to stick to the basics today and try not to get lost in the woods. So here we go. When we talk about genetics and aquaculture, pretty much we're just falling back on the basic principles of animal improvement and plant improvement that have been developed over the last, depending on the species, maybe as many as 1,000 years. A lot of our domesticated animals have been domesticated for maybe 10,000 years. So the simplest thing is just to look at different breeds, different strains, how they differ, which performs better in certain situations. But then when we talk about genetic improvement, we're looking at additive effects. This is the key. This is what I want you to take away today. We got two things that we typically work with: additive effects for selection and dominance effects for crossbreeding. When we cross species, when we cross strains, when we cross individuals, that's all dominance effects. And with some species, we can look at things like developing new varieties, varieties based on hybrids. Finally, there's always a lot of things to keep in mind. There are correlations between growth, fecundity, disease resistance. But that list right there pretty much sums it up. It's not complicated. So let's start out with the simplest things: strain effects. You hear around the world, you'll hear a lot of people talk about, yeah, I grow Nile tilapia. Well, if you're in the United States, most of the Nile tilapia that you might have access to came from these little populations right here. Maybe a few from down there, and maybe just a few from Cote d'Ivoire. But mostly, this is what people talk about. But there are lots of other Nile tilapia populations, lots of strains, if you will. And the same thing's true here in the US with some of our most important species, things like channel catfish. This is a study that was done, about 20 years ago, a little longer than that, over in Mississippi. But the point is, they were comparing different strains of catfish, channel catfish, and of course, none of them varied too much in survival. They take real good care of their catfish over there in Stoneville. But look at the differences in weight gain, look at the differences in feed consumption, and in feed conversion efficiency. You can see there are some pretty significant differences, pretty significant advantages to be had, depending on what strain you choose to work with. And interestingly enough, the superior strain here served as a foundation for a lot more work there at the Catfish Genetics Research Facility. So strain effects are important. And a lot of times one strain that does well in a certain environment will not do so well necessarily in another. So it's something to think about. Selection. Selection is what most people think about, what comes to mind when you think about, oh, genetic improvement. I'm gonna improve my tilapia, I'm gonna improve my shrimp, I'm gonna improve my gouramis, whatever it is. And people, you know, the typical thing we think about is selection. Well, the basis for selection is what we call additive genetic effects. Additive effects are just what it sounds like. They add up. You've got positives, you've got negatives. The sum of those gives you more or less what the genetic value of that individual is. Now, if we could see the genetic value of those individuals, it'd be real easy to decide which ones we want to use for breeding stock. But normally we really can't see that. We've made a lot of advances, and I'll talk about that in a little bit. But here's what we're usually faced with, the curve that you see down here. This gray curve, this gray distribution here, this is what we can observe. This is what we can measure in this population of, in this case, we're talking about some shrimp. But that curve underlying that is the genetic values of those individuals. And specifically in this example, those are the additive genetic effects, the additive genetic values. So you can see, you know, we've got some of these animals way out here that are among the most superior, but only part of that superiority is because of their genetics. The rest of it is genetics that are not inherited, things besides additive effects, environmental circumstances, random variation, what have you. I put this normal curve here because I want you to think about, these are the proportions of a population that usually you will find when you plot out a normal curve. Here's your mean, 1 standard deviation, 2 standard deviation, 3 standard deviations, and down the other way as well. And you can see you don't have a lot to work with if you need a lot of animals to produce your next generation. You've got to come up with some sort of a threshold. And here's a good example of that. If we need all of these shrimp to put into our hatchery, then in this example, we've got a selection intensity, we've got a cutoff point of about half a standard deviation. All of these we're going to keep, all of these are gone. Remember that red X. Now, if we don't need so many, which is usually the case in shrimp because, you know, they make so many babies, we've got a lot more leeway. We can go way up here to 2 standard deviations and just keep these guys over here. And that's fine, but we still have this red X. What that means is if we're practicing selection, these guys are gone. All of their genetics are gone. They're gone. That's the price you pay for selection. In this example, you can see some of the math that's involved here. And it's not complicated. When you hear math, don't freak out. It's not complicated. We've got this population here, and we want to keep the biggest 5% of this population to set aside for broodstock. So this is what we call our selection intensity. Over here, this is the mean of those animals that we're going to keep. And we let them spawn, we let them cross amongst themselves into the next generation. We've done it. We've moved this mean over about this far. That R right there means that's the response. That's how much we've moved that mean over. And about 20% of that population is the same size as those 5% that we selected. But look, the mean got bumped over here. It's not way over here where the parents were. and that's how this usually works. Big shrimp crossed with big shrimp gives us bigger shrimp, but not really as big as the ones that we crossed.
Things like that.
Someone, I think someone should mute. Someone's got to mute. Now, heritability. When we talk about genetics, we talk about selection, you hear this term tossed around all the time. Heritability, heritability. What is heritability? Well, we've already pretty much covered it. When we look at what our selection intensity was, and we look at what our response was, we can calculate the heritability. In this example, we selected these parents, nice superior animals. We let them spawn, and the next generation, boom, no response. In that situation, mathematically, we've got a heritability of 0. Now over here in this example, we've got these selected animals, we let them spawn, and oh my gosh, their offspring has the same mean as those selected animals did. That would be a heritability of 1. This doesn't happen. This never happens because there are too many other factors that contribute to this superiority. Now this heritability of 0, that does happen. That has been reported a lot of times for different traits, for different species. So that's your range. That's your range of heritabilities, anywhere from 0 to 1. It's pretty straightforward. You hear people tossing around this term heritability, and they're trying to make you think they're really smart. You're just as smart as they are. All it is a portion of that superiority that's going to show up in the next generation, that's going to be transmitted. Now, some kinds of heritability are a little harder to figure out and calculate mathematically, but that doesn't mean they don't exist. Like this example with tilapia, a lot of people in a lot of places have made good progress selecting for the shape of their tilapia. And all it takes is a few simple measurements and you can do the same thing. Another example from tilapia with coloration, a lot of places where the market wants a red colored fish, they don't want these black blotches. And you can select for that too. You can make progress with that as well. So that's pretty simply how selection works. It relies on these additive effects that are transmitted to the next generation. If none of that superiority is transmitted to the next generation, and basically your heritabilities are 0 or close to 0. Here's some examples. You know, and some of these are fairly outdated. As time goes on, the math has gotten better and the methods have gotten better, but this just shows you, you know, some of the typical ranges of heritability that have been reported in a lot of different aquatic species. You know, anything from maybe 0.04 to way up here like 0.71. That must have been a really lucky hit in terms of agent maturation, because very few heritabilities are going to be that high in the real world. Here's a bunch of examples from white leg shrimp from Vanamai. And you can see again, heritabilities ranging from maybe 0.2 to 0.4. On a good day, you might get as much as 0.5. This was a study that came out of China. And we like to calculate a standard error with that heritability to get some idea of, you know, just how realistic is it. Here's an example. In recent years, there's been some good heritability estimates generated on tilapia for disease resistance. This is 2 different streptococcus diseases of tilapia. And in the last few months or so, I've seen a nice study on the heritability of resistance to tilapia lake virus. But remember that slide with the shrimp, and we've got that curve of the gray values that we observe, and then we've got that underlying curve of the genetic values of those animals. Well, heritability, like I said, is just the portion of that superiority that's going to be shown in the next generation. And here's a good example. When we've got shrimp that aren't challenged, that are in a good, healthy environment, they were able to calculate the heritabilities of about 0.15 to 0.33 of that. That's how much of that superiority that we could observe would be transmitted to the offspring. Now, if you throw something else into the mix, which is infect that population with white spot virus, suddenly that heritability estimate goes down. And that's because there's a lot more things going on that influence the superiority that we can observe that may have nothing to do with growth in total weight. So it's just a good illustration of the fact that heritability, it's all relative. And there's no pun intended there. It just depends on the population you're working with and the way you're raising those animals. So selection, I'll say it one more time, is based on additive effects. It's based on the genetics that are transmitted directly from an individual to its offspring. And now you guys understand more or less what heritability means. If your heritabilities for the trait you're trying to improve are like 0.2 and higher, mass selection is the best, most efficient approach to genetic improvement through selection. Mass selection just means you got a bunch of animals and you pick the best ones, and you're not worried about their pedigree, you're not worried about what family they came from. You got a bunch of animals and you pick the best ones. Now, if your heritability is low, like less than 0.15, or if you're looking for the heritability, say, of dress out percentage, and once you measure dress out percentage on an animal, you're not going to be able to spawn it, then family selection might be preferable. Families have to be marked, they have to be traced back to their families. You got to know what family an individual came from. If you have to keep them separated to do that, then it gets pretty complicated and you get a lot of outside factors that can bias your estimate. It works better with big families, but that doesn't mean it's preferable for big family species. And a lot of other factors are going to be involved in that too. So you want to do selection on your farm. Maybe it's a big farm, maybe it's a tiny farm. Either way, when you get started, you're going to have some costs involved. Let's say this line right here is your steady state. This is, you know, more or less the money that you're making from month to month or year to year in your farm. Now you're going to start the selection program. You've got these costs involved. More costs to get set up and started, and then some cost from that point on to do your selection program. But you're also going to have a lot of gains. You're going to have gains in growth, maybe gains in survival, maybe gains in feed conversion. So you're going to have these gains and you're going to have these costs. So let's add them together. And this little line right here is pretty much the sum of your costs and the sum of your gains. A lot of people get all excited when they reach this point right here because they think, oh my God, you know, I'm back to where I started and now it's all gravy. Well, not really. This is the point in time that you need to be thinking about when all of the gains offset all of the costs. And from that point on, then you could say, yeah, it's gravy. So that's the way you need to think if you're going to invest in some sort of a genetic improvement program on your farm that involves selection. That's all I'm going to say about selection. Additive effects transmitted across generations, different ways to do it. Now, the other big genetic effect that we can take advantage of in aquaculture is dominance genetic effects. Dominance doesn't have anything to do with transmitting something from an individual to its offspring. Dominance is all about combinations. Combinations of specific parents, combinations of varieties, and even combinations of species. That's what we talk about when we talk about hybrids. Dominance effects with species. And this is how it works. It's really simple. You've got one line, you've got another line. If everything was just additive, this would be the performance of the offspring. But quite often the performance is above that. And that's what we call heterosis. A lot of people call it hybrid vigor. This is used a lot in plant breeding. Here's an example from corn. They like to generate lots and lots of different lines of corn. And, you know, a lot of them are really not suitable for commercial production, but when you cross them, you get a really nice hybrid. That's why you hear people talk about hybrid seed. It's simple, it's easy, it's just combinations. We see this a lot in aquaculture in some parts of the world. People are still taking advantage of a cross between blue tilapia male and Nile tilapia females. This is a big deal 30, 40 years ago. You get a nice hybrid animal, and quite often you get a high portion of males. In the United States, this has really made a comeback in the catfish industry. People are crossing channel catfish with blue catfish to produce a hybrid. It's got a lot of superior traits in terms of production. Here's just a little picture of some little baby hybrid catfish. They're not as easy to produce as pure channel catfish, but the performance offsets that cost, that investment. And of course, a hybrid striped bass, another classic example of how we can use dominance effects in aquaculture. Here's an example. I had a friend many, many years ago who ran the hatchery for a big tilapia farm. And of course, they were harvesting 20 to 30 tons of fish a day, maybe 25 days a month. So he needed to make lots of babies. He had a male line. The idea for his male line was these blue tilapia are more cold tolerant. The red tilapia are more docile, easy to handle. He crossed those to get his male fish. Females, he needed a fish that would produce lots of offspring. So he crossed two distinct lines of Nile tilapia, and he got these fish, and he was able to bump up the fecundity on these fish because they were crossbreds, heterosis, and it worked for him. Now they filleted all their products, so they didn't really care what color they were. They could be red, orange, black, blue, didn't matter. Spotted, freckled, didn't matter. And this system worked really well for him. And again, he was focusing on using dominance genetic effects, using combinations to get the fish he wanted. Now, dominance has a dark side too, and that's inbreeding. Dominance effects are pretty much the basis for what we see when we see inbreeding. Everyone hears inbreeding and they think, oh, that's bad, that's bad. Well, it's not necessarily bad. And here's two examples. Santa Gertrudis cattle, they're all over the world. They're really highly prized breed. They're really good at what they do under certain circumstances, and that entire breed can be traced back to one bull. His name was Monkey. And also German Shepherd dog, used in police and military operations all over the world. They're the second most popular breed of dog in the United States. All the German Shepherds in the world, the purebred ones, can be traced back to one male dog. So inbreeding is not necessarily a terrible, terrible thing. Now, should we fear inbreeding? Not necessarily, but we should fear inbreeding depression. And here's a couple examples of inbreeding depression in tilapia. It can cause deformities. It can cause those fish to kind of regress back to a less improved form. But we can control inbreeding depression. We can control the accumulation of inbreeding itself. And here's a good example. Now, this came from a paper back in 2002, and we're talking about mass selection here. You'll hear some people say, oh, well, you got to use family selection. If you use family selection, you can eliminate inbreeding altogether. And I could talk about that later on, but there's a lot of literature that contradicts that statement. But in this example, we're talking about mass selection. Here's an example where they ran a computer simulation for 15 generations. We're trying to improve a trait that has a heritability of 0.2. You guys remember what that means, right? Heritability of 0.2, that means about 20% of that superiority that we can measure will be expressed in the offspring. Now, each of these simulations here had 4 pairs of brood stock per generation. And well, in this one, we had 4 pairs of brood stock per generation. Each of them had either 5, 50, or 150 offspring for each spawning, each mating per generation. You can see if you've only got 4 pairs of brood stock after 15 generations, you know, you've got a pretty nice selection response. You've improved that trait by almost 80%. But look how much inbreeding you've accumulated, up around 70%. This is with only 4 pairs of brood stock. That is what we would call an effective population number of 8 animals. Now, if you bump that up to 100 pairs of brood stock with the same numbers of offspring, you look at your selection response. You're way up here around 130%, but this is the important part right here. Your inbreeding has only accumulated by maybe as much as 10%, which is easily offset over 15 generations. So you can have selection, you can have genetic improvement, and you can keep inbreeding under control. Nonetheless, this is the truth. Remember that X. All of this is gone. There might have been some real gems in here, but you'll never see their offspring because it's all gone. That's just how selection works. I mentioned effective population number in those 2 examples. This is what we're talking about. It's just a mathematical concept. It's mathematically the number of animals you have contributing based on the number of males and the number of females. And here's a real simple example. You got 2 little guys that have their little hatcheries out behind their house. They both have 30 brood stock. This guy has 25 females and 5 males. His effective population per generation is only about 16.7. This other guy has 30 animals, but he's got 10 males and 20 females. His is up way closer to 30. Now, he's not going to produce quite as much fry, but he's also not going to accumulate inbreeding nearly as quickly. How do we figure out what the heritability is besides just, you know, breeding some animals and seeing how their offspring do over time? Here's a quick way. We use statistics. In each of these little boxes here, we've got 1 male and 2 or 3 or 4 females. And we compare the variation in the performance from all these different families. And here's an example. The offspring from this female, what do they have in common? They have their mother and they have their father. That's what they have in common. The offspring from these 2 females, what do they have in common? The father. So what they have in common here, we're not talking about combinations, we're only talking about what they get from him is what's transmitted directly, which is additive effects. Now, what do these guys have in common and these guys have in common? Nothing, not the father, not the mother. And with statistics, you can take the performance of all those families and you can see, just relatively speaking, how important the male is versus how important the combination of male and female is. I had a few people try to get in touch before this started. There's some other things that we hear about a lot in genetics and aquaculture. One of them is ploidy manipulation, people trying to produce triploid animals. We could talk for 2 days about this, but basically it's pretty simple stuff. We're going to start with a normal fish. This is how it works in fish anyway. When the sperm fertilizes that egg, there's still 1 extra set of chromosomes in that egg, and it gets pushed out. That's called the second polar body. We're not going to talk about the first polar body right now because we don't need to. So the chromosomes from the sperm enter the egg. The chromosomes that are in the pronucleus are there. That polar body it's gone. The 2 sets of chromosomes merge. You've got a nucleus now, and everything starts. The chromosomes double, the cell divides, and you're on your way to producing a normal organism that has 2 sets of chromosomes in every cell. If you want to make a tetraploid, you let that process proceed, and then right about when that cell is going to divide, you hit it with a shock. It could be a pressure shock, heat shock, cold shock, could even be some nasty chemicals, and you stop that separation. So now you've got a cell that's a little bit bigger, and it's got 4 sets of chromosomes in there. You can do the same thing with triploids to make triploids, except you apply that shock right when that polar body would have gotten kicked out. It doesn't get kicked out. So now you've got a nucleus with 3 sets of chromosomes. And from that point on, it develops pretty normally. The thing is, if you want to produce a lot of triploids, you got to do this for every batch of eggs. The alternative is produce some tetraploid animals, cross them with normal diploid animals, 2 sets of chromosomes, and you can mass produce triploids, which is pretty much the way people like to do it. Now, I'm going to go back. The timing of this shock and the timing of this shock are critical to whether you're going to be successful or not. So people do a lot of this work in labs. They'll fertilize eggs at different times. Then they'll hit them with a shock, a cold shock, a heat shock, what have you. Incubate the eggs. This is some work we did in my lab many years ago. And then see what the timing is that produces the most triploids or the most tetraploids. You see a lot of talk about triploid mollusks also over the years. It's a little more complicated in mollusks, and I'm not going to go into the details right here, but you know, it's not rocket science. Lots of people have done it. It can be done. And again, for things like oysters, if you want to mass produce triploids, you're not going to go through this whole procedure with shocking each batch of eggs. You're going to figure out a way to produce tetraploid organisms, cross those with normal organisms, and then you can produce triploids in massive numbers, especially if you're working with oysters. Then you really are talking massive numbers. Monosex production, it's another part of genetics that people get excited about. It's pretty easy to do in salmonids. You can make all female salmonids. People have struggled with this for years in tilapia, trying to make all male tilapia. The idea though is you're using things like estrogens to produce an XY animal that functions like a female. Then you get some XXs, you get some XYs, and you get some YYs. And then you got, in the old days, you had to do progeny tests. You had to cross all these animals with normal animals and figure out which ones were YYs. Now we have something called marker-assisted selection. It makes the process quite a bit simpler. You don't have to do all these progeny tests. And that's all I'm going to say about that. Marker-assisted selection, though, I do want to talk about. How does that work? Well, it's not always easy to trace genes to their specific locations on chromosomes, but it is a lot easier to trace markers. And there's different ways you can do this. There's lots of different kinds of markers. Here's just an example. You can take some of these markers and get like a fluorescent dye and figure out exactly where they are on the chromosomes you're looking at. A lot of work has gone into this to develop maps of chromosomes. Here's how markers work. Here's a little animal we got here. He's in this pair of chromosomes, this chromosomes here that he's got. You know, he's got one that came from his sire, from his dad. He's got one that came from his mother over here. These were the 2 that dad had. These are 2 that mom had. It's pretty straightforward over here on this side. This one right here is what was inherited. But this one is not like either of these that the sire had. And so what happened? How did this green thing get associated with this gray thing when they were on 2 separate strands over here? We had crossing over. This happens all the time with strands on chromosomes. They cross over and they connect with the parts of the other one. Now, crossing over, like I say, happens all the time, but this is where the markers come in. The closer 2 points are on that chromosome, the less likely crossing over will occur in between those 2 points. You got all this area for crossing over to occur, but it's going to be a rare instance where it occurs between these 2 points. The closer they are, the less likely it is to happen. And that's where the markers come in. You may not know a lot of specifics about this gene, but if you can track that marker, that's mostly all you need to know. And that's how markers work. Now, the science of markers has really advanced in recent years. It's led us to a point now where we can get lots and lots and lots of markers for lots and lots and lots of genes that we believe are associated with the traits we're interested in. And then we can look at all of those markers at the same time and try to sum up the good genes and the bad genes and come up with a breeding value for the animal that we're looking at. Now, it sounds real complicated, and it can be real complicated, but the basics are the same. We're still back to trying to figure out what the genetic worth is of an animal by some kind of way that we can measure it. One of the things that they use a lot now, they're called SNPs, single nucleotide polymorphisms. They go in there now with sequencing, and they can look at the whole list of the whole length or sequence, if you will, of all the little letters. I'll just call them letters. All the little letters on a strand of DNA. And you will find different forms. They're almost identical, but you'll have one little change, and that's why they call them single nucleotide polymorphisms. And those are really useful as markers to try to find the genes that have a big influence on a trait. Like, here's an animal, it's got all these chromosomes here, and they've been able to link certain SNPs, certain markers with certain genes, and see how much they affect the trait that they're interested in. You can see most of them don't have much of an effect at all. A few of them, a little bit. But on this chromosome 14, you've got a couple of these genes that have a pretty significant effect on this trait. And I won't go into a lot of details, but that's the direction we're going. In terms of molecular techniques, it's gotten a lot more complex, but it's still the basics. It's still what made this fish so big. How much of that was his genes? How much of that was genetic effects that could be inherited? That's what we're still after for the most part. With all these new advances in molecular techniques, we're still trying to say, how big will his offspring be compared to his offspring, and why? So that pretty much covers the basics that I wanted to talk about. I guess we're about 40 minutes into this now, and I would like to go back to my hosts and see if we can talk about some of the questions that have been asked.
So one question was, why is heritability not as high as 1 in the real world?
In the real world, a heritability of 1 would mean that no other factors were involved in the performance for that trait. No dominance effects, no environmental effects, no maternal effects, no random effects. A heritability of 1 would mean that additive genetic effects were responsible entirely for the performance that we could observe. And every trait that we have any interest in is controlled by at least a few, and sometimes as many as 20 or 30 or 50 or even more genes. And so that's why if we get a heritability of 0.2, we should be happy. If we get a heritability of 0.5, we should be ecstatic, because that means we've got a lot to work with in terms of improving something through selection. We will be able to see a lot of that superiority reflected in the offspring.
Another question was, are heritability and breeding value the same thing?
That's a good question. Heritability is involved in the breeding value. You could say that the breeding value is a reflection of that animal's genetic worth in terms of what it will transmit to its offspring. Heritability just means the portion of superiority that we can expect to be transmitted. Breeding value means the performance that we could expect from the offspring of an individual if it was crossed at random with the rest, with other individuals from that population. The breeding value is if you can still see my screen here, it is where we would expect that animal to fall out on this dotted line here. It is the genetic worth of that animal. And so you may have an animal that's, you know, not mediocre, but just average, but it actually has a really good breeding value. It's just that it's had some bad luck or other factors that push it down to right here. So heritability is more to do with how much of that superiority you would expect to see transmitted. Breeding value has to do with that specific animal and what you would expect its offspring to perform like.
Next question, can relationships calculated using molecular markers be used to calculate heritability?
To the extent that you had enough molecular markers to look at the whole variation for that trait, you could probably calculate heritability, but it would be an awful expensive way to do it. You would have to say, okay, we're gonna look at the genomes of all these animals in this population, and from that we are going to calculate a distribution of the additive variance in the population. And we know what our phenotypic variance, the variance we observe, is. And so then we've got that ratio again, which is, that ratio is what you use to calculate heritability. So you could do that, but right now it would still be an expensive way to do it.
Are there different selection programs and mating designs used in selection? Or maybe they're asking for examples of selection programs and mating designs used for selection.
Yeah. This is what's called a nested design. You can also do this the other way, more set up like a factorial design where you've got, if you can take the eggs from the females and cross them with different males as well, then that's a whole nother design that you can use. If you've got that luxury, you can make much more complicated designs. Now, in terms of different selection programs, the big ones, of course, are mass selection, where you judge that animal based on its own characteristics. And you've got family selection, where in family selection, the idea is you've got what you can see, which is gray, and what the genetic worth is, which is this black line. And the bigger the family is, if you've got a characteristic that's got a low heritability, you can get more information by looking at all the members of the family, and their mean will come closer to what the real genetic mean is if you've got a low heritability. It still has a lot of problems, and some of those problems are, you still get a lot of inbreeding with family selection. If you want to delve into the statistics and the quantitative math, only half the genetic variation is expressed between families, so only half of it can be utilized. So it's inefficient in that way. Like I said, the larger a family is, the more closely the average of the values will reflect the average genetic worth of that family, but still there's no way to know which ones are superior or which ones are inferior. You need to maintain a lot of families. You've got to be able to look at a large portion of the individuals in each family. And so, you know, if breeding space is limited or it's hard to make single pair matings, which it is like for some tilapia where they're really aggressive when they spawn, family selection is not necessarily efficient. There are some situations where it's the best choice. So you've got family selection, you've got individual selection, you call it mass selection. You also have things called within family selection. That's when you have to keep your family separated. There are a lot more ways to do it. And you could probably teach a whole college class just on selection and different ways to approach it and different ways to approach the statistics involved with it too.
Okay.
But I'm supposed to stick to the basics today.
Yeah, right. This question is, hi, Dr. Lutz. I'm a journalist, not a scientist. Do you think breeding innovations hold the key for expanding aquaculture? What genetic innovations are in the pipeline that could change aquaculture in the next few years?
Oh, I think definitely breeding is one of the paths that's going to expand aquaculture. You know, we've made big improvements in production systems. We can only go so far with that. We've made big improvements in nutrition, and I think there's still some room to grow there. And then the other one would be genetics, genetic improvement. When we look at traditional livestock, we haven't come anywhere close to the gains that have been made in traditional livestock. And I think we're still talking about selection, but when you talk about genomic selection, the whole approach of having the markers available now to look at lots and lots of genes at the same time, we will be able to quickly catch up with the advances that have been made in other kinds of livestock. I think over the next 20 years, we'll see tremendous gains.
All right, thank you. Next question is, if you bring in a gift tilapia from one country where it's doing well and take it to another country where the environment is perhaps different, will that gift species perform differently in the new environment?
Well, if it's a culture environment and the culture conditions are similar, then it probably will perform comparably. You know, if you've got similar temperatures, similar feed available, similar stocking densities, similar water chemistry. But if you take that gift tilapia and just throw it out in the wild, more often than not, it will not do so well, because the only advantage it really has is fast growth. And you're only going to be able to have that fast growth if you get all the food you want every day. There's a real good study years and years ago, but it was sort of overlooked, but it's still a classic study of it was done in Egypt, and they had 4 different lines of Nile tilapia, and they looked at what we call genotype environment interactions. And they raised those 4 lines in 4 or 5 different production situations. One was in rice paddies, one was in cages, I believe. And it was astounding, the difference in ranks. What works good in one environment doesn't necessarily work good in another.
It says, why don't we use tetraploids instead of crossing to obtain tetraploids? Are tetraploids unstable?
I don't think tetraploids are inherently unstable. In fact, a lot of modern species that we work with were the result of natural tetraploidy, thousands, millions of years back. I don't know that a tetraploid in and of itself has a lot of production advantages. So you would have to take into account the cost. Also, you're not going to produce, if you're going to produce a line of tetraploids, they are probably you're going to have to cross tetraploids with tetraploids, which means you don't have the luxury of starting out with lots and lots of numbers. So you may be at a disadvantage already in terms of inbreeding accumulation. Now, like I've said, we don't have to be afraid of inbreeding, just inbreeding depression, but that could be a disadvantage as well. I think the reason people are interested in tetraploids is almost always to be able to produce triploids. You know, I will give you an example in terms of instability. That can be the case sometimes. I had a student who struggled to produce tetraploid tilapia, and we used flow cytometry, and we knew that we had produced tetraploids. And over the course of 4, 5, 6 weeks, they all just disappeared. And later I read a paper from a really outstanding research group in France. They had produced tetraploids. And you have to remember that cell is bigger and the nucleus is bigger. And what was happening was the blood cells were so big in these little larval tilapia that they couldn't pass through some of the arteries and veins and blood vessels. And they were piling up and clogging up, and that's why those larvae were dying. Of course, it was 6 or 8 or 10 years later after we had given up on it that I read that. But that is one of the inherent things that you could run into trying to produce tetraploids.
Thank you. Let's see. Next question. How has genetic work changed over the last 5 years? For instance, in terms of the cost, accessibility, and user friendliness, can a mom and pop operation do it on their own with the help of kits, or is this something that remains the domain of laboratories?
Well, that's, you know, I hate it when everybody says, that's a great question, but that really is a great question because it touches on a lot of issues. There is definitely a technological divide beginning to become apparent in terms of genetic improvement in aquatic species. There are still a lot of things that mom and pop can do on their farm in terms of how they manage their breeding stock, how they do their selection, or if they want to cross distinct lines. There are a lot of benefits to be gained without having to use all of these molecular methods. I look at the poultry industry a lot when these questions come up, and I don't have a good answer for them either, but if you look at what's happened in poultry over the years, you know, you've got two entirely different things. You've got commercial lines of poultry, and there's just a handful of organizations or companies that pretty much control that. And then you've got, you know, your mom and pop, they've got 8 or 10 chickens that run around the yard. You could not take one of these commercial poultry house chickens and throw it out there in the yard and expect it to do very well. And vice versa, you couldn't take those yard chickens and throw them in a poultry house and expect them to do very well. And this is a reality we're going to have to deal with. It is emerging in aquaculture, and we may get to a point where all the good fish are going to trickle down from these breeding companies, and that's what everyone else will have to work with. I don't think we're close to that yet, and I think there's, like I said, there's a lot we can do on the mom and pop scale while that other advancement goes forward.
Here's a related question. Why using YY tilapia cross or male tilapia cross with females? Will we still get females in the fingerlings? It's supposed to be all males. Is it a genetic error? Is the farm manager question.
This is why this YY technology, for all of its promise, is still not widely adopted. there is another major gene that influences the sex, apart from the X and Y in Nile tilapia, and the W and Z in blue tilapia. There's another pretty major gene, and research would suggest that there's as many as 3 minor genes that are involved. And then you've got aromatase, you've got temperature-related influences. You can have a pair of tilapia that will produce 100% male offspring under certain conditions, and under other conditions, it may be as low as 60% or 50%. There are just a lot of factors involved besides that simple XY system. And that's why it's been so hit or miss.
Well,
Okay.
again, we appreciate everybody coming, and we will try to announce, we plan to post the recordings of these webinars. We may try and clean them up a little bit, but we plan to post them on the United States Aquaculture Society YouTube channel, as well as my Aquaculture Education YouTube channel. And hopefully we'll also be able to link it to the National Aquaculture Association's website. But I think we'll go ahead and wrap it up. We certainly appreciate everybody, and you in particular, Dr. Lutz, for sharing your information with us today. And we appreciate everybody for coming.
Thanks, everyone.